So the next special sense that we visit is hearing. And so we've seen some already that are photoreceptors. We've seen some already that are chemoreceptors, that's including taste and smell. And now we are getting to those special senses associated with the ear, which most people would have known was hearing, but we also said equilibrium is our last one. So we're going to start out with hearing, and we're going to look at the anatomy like usual, look at the structure, and then we'll talk about the function that comes from that structure. And what we're going to find is as we get into the ear, 3 different regions to it, and when we get to the receptors, whether it's hearing or equilibrium, we're going to find that these are going to be mechanoreceptors. So remember, mechanoreceptors have to have physical deformation in order to send a signal into the brain. All right, so when we look at the ear, we have 3 different sections. We've got the external ear, we've got the middle ear, and then we have the inner or internal ear. And each one has their role to play in terms of particular parts, etc. Now when we look at it, particularly, we'll go back here, as we navigate our way through, right? So the sounds are going to be passing into here. As well, your Q-tip might be passing into there. It's not supposed to because it could impact this structure, the tympanic membrane. But in any case, we have our auricular pinna, which acts as a funnel to be able to gather the sound waves. And then it's going to route it down the external acoustic meatus, which is also known as the external auditory meatus. We saw this when we were looking through the skeletal system, right? The temporal bone. And so that's going to lead us into the middle ear. So the middle ear contains the ossicles, the different ear bones, and we have the tympanic membrane. So the tympanic membrane is kind of the border between the outer ear and the inner ear. So as the sound waves come in, they're going to cause that tympanic membrane to vibrate. And as that vibrates, then the different ear bones are going to move as well. So we have the malleus, the incus, and the stapes. Right? Once upon a time, the hammer and the anvil and the stirrups. So these are going to transmit the signal into the inner ear. They're also going to amplify that signal. And when we look in the inner ear, we see that we've got a snail-like structure that's called the cochlea. We also have structures over here that are separate and distinct. Now, while the cochlea is associated with hearing, the semicircular canals and the utricle and the saccule, which are contained down in an area down here, which is called the vestibule, are associated with equilibrium. And then we can see the nerves that are taking these special sense stimuli into the brain. So remember, from central nervous system and then peripheral nervous system, we have cranial nerves. So cranial nerve number 8 is the vestibulocochlear nerve, right? And we can see that there's 2 branches from it, going to the vestibule, going to the cochlea, routing that information from those sense organs into the brain. Now, one of the other things we see as we're looking at these structures in these 3 different areas is we see the pharyngotympanic or auditory tube, once upon a time also known as the eustachian tube. See how it leads into the middle ear there? That's very important for us to be able to balance the pressure on either side of that tympanic membrane. And so when you push air up into it, so whether you talk about going up in a plane or going down scuba diving, grab your nose and blow air to the back of your pharynx, and that forces it up into the pharyngotympanic tube, and it equalizes the pressure on either side of that tympanic membrane. So when the pressure outside in that external acoustic meatus is higher or lower than the pressure in that middle ear, that's going to cause the tympanic membrane to flex out or bend in, and we want it to be normal, and we want it to be flexible. And so if there's too much pressure on either side, we need to equalize that pressure. And so that's what we do. So you're going up in the plane, you're chewing gum, you're blowing into your tympanic membrane on the opposite side of it, in that inner side of the middle ear, and that's going to equalize it. So we can see that coming up in the lower left-hand corner right here. So that's where that air is going to enter. We can also see it coming up through there. We can see those ear bones, malleus, incus, and the stapes, as we mentioned, on the other side of the tympanic membrane. You'll also notice some muscles. So here's a tensor tympani muscle. There's also the stapedius muscle that we see right here. So these muscles work to protect the middle ear structures, the bones and the tympanic membrane, to make sure that under extreme intense sound waves that they're not going to be damaged by moving too much. So they restrict the movement of them, kind of dampen the movement of them so that they don't get damaged. In fact, remember when we were going through the skeletal system, we said the stapes was the smallest bone in the body. Well, the stapedius muscle is the smallest muscle in the body. Okay, so we move from the middle ear to the inner ear. We saw a variety of structures there. Now this is all located in the temporal bone. And so this is in a thicker portion of the temporal bone that's called the petrous portion. And there's a bony labyrinth in there. There's a carved out area, if you will, that allows this membranous structure of both the semicircular canals, the vestibule, and then our cochlea to exist within this carved out area. So we'll look at all three of those structures coming up. Remember that the cochlea is associated with hearing, and then the vestibule and these semicircular canals are associated with equilibrium. So we'll start out with that vestibule just as a tour, looking at the anatomy again, and then we'll get into that physiology. So like I mentioned, the vestibule is like a small room, right? So sometimes you hear about a church or something like that, a big building, there's a small room in one area of it, that's the vestibule. So this is also a vestibule, and it contains, as I mentioned, two sense organs, the saccule and the utricle. And we'll see those coming up in terms of their particular function. Semicircular canals you saw, they're not entire circles, so they're semicircular canals, and they're organized in three different planes, like the X, Y, and Z planes. And so those have fluid inside of them, and we'll talk about how they contribute to our equilibrium as well. So both the vestibule with the utricle and the saccule and the semicircular canals are giving us our sense of equilibrium, but slightly different types. Then we have, like we said, the snail-like cochlea. So we'll look at the inner structure of the cochlea coming up and focus on hearing first and then wrap up with equilibrium. So if we look closer at the cochlea and we look inside of these snail-like or cinnamon roll-like coils, we can actually see that each of the coils is divided into three. It almost kind of looks like a peace sign, if you will. So you can see how it's kind of divided like this. So there's three different areas that we see through here. And so we'll take a closer look at that. One individual coil surrounded by the bone. So we can see that there's going to be two dividing lines, one here and one here. So the upper one is the vestibular membrane. The lower one that we see here is going to be the basilar membrane. This kind of forms the base underneath what we call the spiral organ. Now remember, this is just a slice through this coil that's going all the way up to the apex. And so these tubes are going all the way around. That spiral organ is also spiraling around. So that's where it gets its name. All right, so we have two dividing membranes, like we said, a spiral organ, and then we have three different chambers. We have the scala vestibuli, which is near the vestibular membrane. We have the scala tympani, which is near the basilar membrane. And in the middle, we have either the scala media, right, in the middle, or the cochlear duct. And so the other two, the scala tympani and vestibuli, both contain perilymph, a fluid, and then the cochlear duct has endolymph instead. And the fluids actually conduct sound waves a bit better than air. So for instance, if you were banging on something, that's probably pretty loud since that's my microphone, but if you were banging on something in the air, you could hear it for a certain distance, but if you're doing that same percussive movement underwater, you could hear it much further away. So in fact, when you're scuba diving, in order to get somebody's attention, you take something metal and bang it on your tank, and that can be heard very far away. Okay, so we're seeing the anatomy here inside of the cochlea. Now we're zooming up closer on that spiral organ, and we can see a few structures associated with that. So orienting ourselves, we have our basilar membrane again. Here is our vestibular membrane cutting off at an angle. But notice with that spiral organ, we have this tectorial membrane that's sitting on top of it. And so the spiral organ is made up of a few different types of cells. We have supporting cells, we have outer hair cells that we see out here, and you can see how they have little hairs on the apical surface, and then there are inner hair cells in here, also with little hairs projecting out. So with those hairs, we're going to find those cells, outer and inner hair cells, are going to be our mechanoreceptors. And so what we find is that with that tectorial membrane impacting those hairs from those hair cells, when a sound comes in, and we'll see this coming up, the basilar membrane is going to bounce. So imagine, I always think of a bunch of kids, little kids bouncing on the top of a bunk bed, and there's a ceiling up above, and so they start bouncing, and if they bounce high enough, then what's going to happen is, right, they're going to impact that ceiling, and their little necks are going to bounce. They're going to flex like that. Not to where they're going to hurt themselves, but in a way, right, that tectorial membrane being right there, when that basilar membrane bounces with sound waves going by, then those hairs on those hair cells are going to flex. And when they flex, like we said, kind of like an ice cube tray, that's going to open up channels, and that is going to then basically stimulate that cell and give us our potential, our membrane potential change that's going to then relay as a signal into the peripheral nervous system, that vestibulocochlear nerve, and into the central nervous system, into the brain. So in fact, you can see here in this scanning electron micrograph, you can see those hair cells. So you can see inner hair cells here and outer hair cells out here. All right, so how does this hearing work? Now that we have the anatomy, we have the external and the middle and the inner ear, we've looked at different receptors for equilibrium and for hearing. How does the reception of these sound waves take place? So, sound waves. So we talked about, when we were talking about light, that occurs as photons, as a particle, as well as occurring in a wave. And we found that wavelength was pretty important, right? Different colors or different wavelengths, and wavelengths are going to be important here. So we want to know, be able to discriminate between different wavelengths, and we'll talk about that, and then also wave heights or amplitude. So when we have a sound, basically, like we said, sound waves, it's this alternating compression and then expansion of particles in that substance. So, and again, if we're talking air, we have molecules in the air that are bouncing around, and we can kind of compress them, and then they expand again. And in water, there's even more molecules, so more compression, more expansion, hence why sound waves travel even further. So remember, when we were talking about waves, right, wavelength is going to be from one point on a wave until we get to that point again, like up here to there, right, to the top of the wave or the trough or wherever you want to measure it, from one point to where it occurs again. That's wavelength. And then, so it could be the crest or the trough, and then amplitude, like we said, is going to be height. So we have width and height. Both are important. So when we talk about wavelength, how short or long that wavelength is, right? Shorter wavelengths occur with higher frequency. Longer wavelengths, lower frequency. And so when we're talking about frequency and wavelength, that has to do with pitch, right? So pitch or tone corresponds to frequency. So high pitch is high frequency, and low pitch or low tone is low frequency. High frequency, low frequency, right? So we need to be able to discriminate those. And so we have a range of about 20 to 20,000 hertz. So how many waves we get per second. Now, that's going to change through time as well, right? As you get older, that range is going to shorten. So particularly those high frequency ones aren't going to be as easy to discriminate. And so now you hear about those apps, right? Those apps that will play high frequency sounds that can be picked up by younger people and not so much by older people, right? It's like kind of like a dog whistle thing, right? Dogs have a higher range of frequencies that they can hear, a wider range, including higher frequencies. And so we blow that dog whistle, nothing, right? They really start perking up. In any case, frequency corresponds to, like we said, pitch. So higher frequency or lower frequency, we talked about already. So amplitude, the height of those waves, corresponds to loudness. So the taller they are, the louder it is. So this is normally measured in decibels, so dB. And so when you get up like above 100 or so decibels, it's actually really intense, really bouncing that basilar membrane. And so that's where some of our ear structures, some of our hearing structures can actually be damaged. Okay, so for these sound waves coming in, we talked about it already, coming in the outer ear, making that tympanic membrane bounce. Then our malleus, our incus, and our stapes transmit and amplify that signal into the cochlea. So where the stapes connects, there is what's called the oval window. It matches the shape, right? Oval window. And so that's going to beat out the rhythm on the oval window, and it sends it into that coiling cochlea. And so the sound waves are going to start at the lower coils, and then it's going to go all the way up to the apex, and then it will kind of coil its way out. And so to make sure that these sound waves aren't just bouncing around in there again and again and again, there's actually an outlet as well. So whereas the oval window is the inlet for these sound waves, there's a round window which is the outlet. So this is where these sound waves can exit and go and dissipate into the middle ear cavity. All right, so why do we have to go up this whole coiled structure? Why can't it just be like one area where we receive it, where those receptors are going to be bounced? Well, what happens is, as it goes through that area, those different hair cells have different levels of stiffness, which means that they will be receptive to different frequency sounds. So for instance, when we're near the oval window, they're very stiff and they're very short, and so high frequency waves are going to only be able to, those are high frequency waves, are very intense waves. And so those are the ones that are going to be able to move those short, stiff hair cells. Whereas when we look at the cochlear apex, when we're getting all the way up to the top, that's going to be receiving those low frequency waves. So the low pitch ones are going to cause the hair cells to bend when we're up at the apex instead. So when we look at it, if we were to unroll that coiled up cochlea, what we would find is at the base, like we said, down here, that's where we're going to have the high frequency, and then those are going to be picked up, and then low frequency are going to be picked up towards the apex instead. So that coiled structure allows us to be able to discriminate different levels of pitch. And then loudness, right, is intensity, how much it's bouncing, etc. Now back to that spiral organ and these hair cells. We said we had outer hair cells, and then we also have inner hair cells. So why do we have two sets of hair cells? And how do those hair cells do their job? So like we said, with the tectorial membrane over them, those hair cells, when the basilar membrane bounces that they're sitting upon, they're going to tend to flex. And in fact, there is a longer one that we see here, and then there are shorter ones as well of these hairs that are called stereocilia. And so what ends up happening, like we said, like my fingers bending, where one moves, it's going to cause the next one to move, cause the next one to move, etc., opens up cation channels that then depolarizes the cell and causes more neurotransmitters to be released. More neurotransmitters, the cochlear nerve, going to stimulate it, greater potential, threshold, action potential, send the signal into the brain. Okay? So that's what's occurring with our hair cells. Now, difference between outer and inner hair cells. The outer hair cells have two major functions. They fine-tune the responsiveness of those inner hair cells by amplifying the motion, but they also can protect the inner hair cells by decreasing the motion. So they're like the taller, if you will, kind of kids in that analogy of them bouncing on the bunk bed, going, okay, all right, you guys are getting near, so I'm going to kind of get some extra stiffness in here to protect you guys so that you guys don't bend your own necks. So, in any case, the hair cells are mechanoreceptors, and those are what are picking up the sound waves and transducing them into action potentials. All right, so once we do that, once we change it into an electrochemical signal, where's it going? So it's going to be routing in. There's a ganglion, right? Bipolar cells, etc. Going in through the medulla, going up through the midbrain, and going to, what do you know, the thalamus, the routing center, right? That thalamus is going to route it out to what we find is the auditory cortex in the temporal lobe. So this is one of the cases, kind of like when we were talking about olfaction and gustation, that the area of the cerebrum that's processing the sensory information is right near the sensory organ that is receiving it. Now, we know that's not the case with the eyes, because where's that going? Where are those signals going? Going back to the occipital lobe. But in this case, it makes sense. It goes right into the auditory cortex of the temporal lobe. Okay, so that is hearing. So the next thing that we'll move into is equilibrium, but we'll do that in a separate little segment. So we will be right back to do that.